For decades, biologists assumed DNA polymerases had one job: copy. But scientists at the University of Bristol just shattered that assumption. They discovered these molecular machines can write entirely new DNA from scratch, without any template to follow. They call it DNA doodling. To understand why this is revolutionary, picture how DNA replication normally works. A polymerase enzyme slides along a template strand like a reader following a script. It reads each base and adds the matching partner. Adenine pairs with thymine, guanine pairs with cytosine. No template, no new strand. That was the rule. Until now. Working at the University of Bristol, researchers gave DNA polymerases no template at all, just raw molecular building blocks. Conventional wisdom predicted chaotic, meaningless output. Instead, the enzymes generated long, structured DNA sequences. Not random noise, but recognizable, organized patterns. Biology's copying machine had become a writer. Imagine an artist given a blank canvas with no reference image. Instead of random brush strokes, they produce recognizable geometric shapes. That is essentially what these enzymes did. Given nothing to copy, they still drew ordered structures into the molecular fabric of DNA. Each enzyme has its own characteristic style, its own molecular handwriting. Hence the name, DNA doodling. Across thousands of experimental strands, the pattern was unmistakable. Where random noise would show no structure, the enzymatic output showed repeating motifs and ordered sequences. Simple repeats, palindromic structures, patterned arrangements, all tied to specific enzyme types. The enzymes were not making random DNA. They were writing their own molecular signatures. The pattern depends on two things: the specific enzyme, and the reaction conditions. Different polymerase families produce different characteristic sequences. Change the enzyme, change the molecular handwriting. Adjust the chemical environment, and the patterns shift. This reveals that polymerases possess underappreciated self-organizing properties, built into their very molecular structure. This discovery addresses one of biotechnology's most stubborn bottlenecks. Synthesizing long DNA strands chemically is notoriously difficult, expensive, and error-prone. Standard methods struggle beyond a few hundred base pairs. DNA doodling could bypass these limitations entirely, with enzymes writing long genetic constructs from scratch, without template constraints. The applications span the breadth of biotechnology. In synthetic biology, enzymatically written DNA could build genetic circuits faster and cheaper. Programmable biomaterials could use long DNA scaffolds as structural frameworks. Bioengineered organisms could be constructed with precision-written genomes. And entirely new forms of molecular manufacturing, biological factories writing their own operating code. Perhaps the deepest implication is for life's origins. One of biology's greatest mysteries is how the first genetic molecules arose, before templates existed and before modern enzymes evolved. Self-directed, template-free DNA generation could help explain how primitive genetic systems first emerged from the chemical chaos of the early Earth. DNA doodling may be a molecular echo of life's first acts of self-organization. The broader implications stretch across science. In medicine, cheaper long DNA could accelerate gene therapy and mRNA vaccine development. In computing, DNA data storage gains a new synthesis pathway. In nanotechnology, self-organizing DNA sequences can scaffold molecular machines. In materials science, programmable biological matter becomes reality. We are at a new frontier in molecular biology. DNA is not just a medium to be copied. It is a language that can be written fresh by biology's own molecular tools. As scientists learn to guide and control DNA doodling, the boundary between natural genetics and engineered biological design becomes far more flexible than anyone imagined. The molecular pen is in our hands. What we write with